Hello, my name is Mike Ward. I'm the head of content at Informa Pharma Intelligence. We're here in Vienna at the BioEurope Spring Meeting, where pharmaceutical, biotech, investors all get together to sort of discuss some of the trends that are taking place in the industry and looking for opportunities for partnering and potential investment. One of the hot areas that we've seen in recent years in that sort of drug development space is the interest in antibody drug conjugates. This, however, has proved to be a challenge. I'm joined by Dominik Schumacher, who is the CEO of Tubulis, which is a brand new company created in 2019, based in Munich. You're in this antibody drug conjugate space, so what's the challenge that you've identified that you also think you have a solution for?
At Tubulis, we generate uniquely matched antibody drug and protein conjugates. And what we do is we have a look at the indication or the disease and design, based on the biology that is behind this indication, a perfectly matched antibody and a perfectly matched toxin or payload. And the way that we do this is that we have generated a screening platform that allows us to rapidly do go, no-go decision making for discovery and development of ADCs. And we have generated a development platform in which we can tackle those shortcomings and the problems that have been around in the ADC field using a biology-inspired approach.
Okay, so we'll come into the details of what those specific challenges were, but initially, the platform, where was it initially developed?
So the platform actually derived from an academic project between Leibniz Pharma Institute in Berlin and the LMU in Munich,
right.
And evolved within these two institutes and turned out to be very highly efficient technologies. And then together with my partner, CSO Jonas Helmer-Smets, we decided to take it further and generate Tubulis.
Okay, and are you restricted to any sort of particular diseases, or if not, how have you decided which ones you're going to target in the first place?
So the way we look at it is that we are not limited by indication, but what our focus is to generate an ADC that has high disease specificity from toxin and payload and also antibody. So we take an indication, we try to find perfectly matching antibodies, perfectly matching payloads, to generate a double selective mechanism compared to single selectivity derived from antibody that has been done in the ADC community so far.
Right, so that's the key difference that your approach has from those others.
One of the key differences. The second one is that we got inspired by a function of microtubule biology that actually enables us to add a significant layer of stability in our ADC products. We have the goal to generate, or what we do is we generate antibody drug conjugates with antibody-like biophysical features.
Right, and that particular property, that's where the name Tubulis is derived.
Yeah.
Okay, so as you said, the company was created this year. How has it been financed so far?
We have been able to collect significant amount of non-dilutive funding, over 3 million euros.
Right.
And this is what we have been using to validate our technology and bring it to the point where we are now. And now we are in the process of doing a seed financing.
Okay, so the validation. So what is the proof of principle that or the validation that you've achieved so far?
What we did is we use our technology to generate different ADCs and took candidates in preclinical experiments, and it's a lot of in vivo testing to really show that our technology can provide ADCs with higher efficiency and also overcome the shortcomings of previous or other technologies. One of the main hurdles is that as soon as you add a payload to an antibody, the physical properties of the antibody change, it aggregates, it loses its function, and by our biology-inspired approach, we can prevent that completely.
Right. So the the areas that you are focusing on initially, can you sort of describe what they are?
So currently we are using our platform now to further develop our own pipeline. We have a number of candidates currently going on in oncology, but we are also looking outside oncology because we think that this has very great potential, or ADCs have very great potential outside oncology as well. And yeah, this is what we are currently doing.
So the seed financing and maybe a Series A financing, when you go out to investors, what are you saying that you will do with that money? What will be the outcomes that they can expect?
So we definitely further develop this platform, validate it further in-house, of course, but what we really want to achieve, or what we want to do, is use this platform for our own pipeline establishment, because we want to bring a new level, an extra level of creativity back to the ADC field, and this is definitely what we want to do.
So, but what would a sort of a tangible outcome be? Would it be a sort of a candidate to a particular disease?
Yeah, for example.
And are you able to share with me what that particular disease might be?
Not yet.
Right. Okay. It's always worth asking.
Yeah.
So this is a seed round and a Series A. What's the sort of the timeline that we're looking for to sort of complete those particular transactions and also then deliver the outcomes associated with that?
So, as it's a fairly new founded company, I think right now it does not yet make sense to give any timelines. But our goal is to, with the seed funding, to really scale our process and really be able to quickly make go, no-go decision for uniquely, perfectly matched ADCs and scale this out quickly.
Right. And sort of looking at what you have to do sort of in the validation, what is the scientifically the sort of the biggest challenge that you think you're going to face in sort of the coming 12 months, 18 months as you seek to sort of validate the approach? When will you be able to make your go, no-go decision?
I think, and this is quite good, from the developments in the ADC field in the last years. It's a lot of learning from fallbacks, but also some success stories. Yeah. And based on these, we can really identify critical experiments that we have to do to then be able to actually make decisions. An example would be for, of course, efficacy, how it is working, but also manufacturability, something that also you have to keep in mind when you design an ADC.
Yeah, great. Well, look, Dominik, thanks very much for stopping by.
Thanks.
Great.